Welcome to Generative AI Futures.
Today, we are doing a deep dive into, well, what might be one of the most transformative areas in modern medicine, the place where generative AI meets therapeutic antibody design.
That's right. And we're going to be looking very closely at a system from Nabla Bio called JAM. It's Joint Atomic Modeling, and its much more powerful successor, JAM-2.
Exactly.
And our mission for you today is to go way beyond the headlines. We want to give you a really comprehensive technical, mathematical, and of course biological breakdown of how these models are designing what look like drug-like antibodies.
And we want to see how they stack up. How do they compare to the current state of the art? The big question we're asking is, are we finally at that inflection point? The point where de novo computational design can reliably hand over leads that are basically ready for the clinic?
The opportunity here it's just astronomical. I mean, this matters whether you're deep in biotech or just curious about where medicine is heading.
It really does. Antibodies are already the biggest and fastest growing class of drugs out there. They're used for everything: cancer, infectious diseases, autoimmune disorders.
But the discovery process has always been the bottleneck, hasn't it? It's slow. It's stochastic.
Completely. You're talking about screening billions, literally billions of different variants, maybe from immunizing an animal or using these complex display technology.
Age display, yeast display.
All of them. And at the end of the day, it's a numbers game. You don't really have that fine-grained atomic level control over exactly where your antibody is going to bind. You just hope you find a good one.
The promise of this de novo computational approach, though, is to just completely flip that script.
Right. Instead of sifting through a haystack, the AI designs the needle. It builds an antibody, residue by residue, to hit a very specific spot, an epitope that you, the user, have defined.
So you get radical speed and, maybe more importantly, radical control.
And that brings us to the core claim we have to unpack today. The claim is that JAM-2 has actually delivered on this promise in a way nothing else really has before.
They're saying they can get clinical-grade properties. We're talking picomolar affinity, which is incredibly tight binding.
And strong developability, so it's not just a binder, it's a manufacturable drug.
And all of this straight from a fully computational design. No need for months of tweaking in the lab. That's the, for the first time, claim that we really need to investigate.
The data they've published is, frankly, pretty compelling. You're seeing double-digit success rates across 16 different targets the model had never seen before.
And it hits the hard stuff.
It hits the really hard stuff. Things like native GPCRs, those are G-protein-coupled receptors, these crucial messengers on our cells, hitting them directly on the cell surface. This is the moment where de novo design arguably goes from a cool science project to a real industrial-grade engine for drug discovery.
Okay, let's start with the foundation. To get JAM-2, we first have to understand its predecessor, the first-generation system, JAM. So what is it, fundamentally? What are its inputs and outputs?
At its core, JAM is a generative system for designing proteins. But, and this is the key detail, it's not just spitting out a single protein sequence. Its input and output space is the protein complex.
The complex. So not just the antibody, but the antibody and the target it's binding to all at once.
Exactly. It's handling the amino acid sequence and the precise 3D structure of all the interacting parts together. It's modeling the binding event itself.
That sounds like a much, much harder computational problem.
It is, but that's where the power lies. The way it designs is by generatively filling in the missing pieces. So you give it the knowns, the structure of your target protein, for example, and then you mask out, you digitally erase the parts that you want the AI to create.
The antibody.
The antibody. And JAM generates both the sequence and the 3D structure to perfectly fill that gap.
Okay, but binding isn't like two pieces of Lego clicking together. It's flexible. Things move. How does the model account for the fact that the target itself might need to shift a little to get that perfect fit?
This is a really critical point, and it comes down to the idea of constraints. The system uses two different types. First, you have hard constraints.
Things that absolutely cannot change.
Right. The sequence of your target protein, for instance, that's fixed. But then you have flexible constraints. The 3D structure of the target is a perfect example. You provide an initial structure, but you tell the model, hey, this is allowed to move a bit.
So the model can actually nudge the target's atoms around to find a better binding solution for the antibody it's designing.
Precisely. It can co-design the complex. The final output might have a target structure that's slightly different from the input, because that subtle shift allows for a much tighter, higher affinity interaction. This is absolutely essential for tricky targets like GPCRs, which are naturally flexible.
That feels like a huge step up from models that just design against a single static PDB file. So let's get into the weeds of the architecture. The sources say there are 3 core components.
Yes, and they all work together. It's a system. The first piece is the engine, the creative part. That's the generative model.
The deep learning backbone.
That's the one. It operates on these protein complexes, juggling sequence and structure at the same time. It takes your masked input and fills in the blanks. You can think of it as the sculptor.
Okay, so the sculptor makes something. Then you need the art critic to tell you if it's any good.
And that's component number 2. The scoring model. This is its internal quality check. It looks at a generated complex, the antibody and the target, and it outputs a single score. And that score has been trained to correlate with real-world experimental success.
So it's not just about a good geometric fit. It's asking, is this actually a drug-like molecule?
Exactly. Does it have properties that suggest good affinity, good developability? It's learned the subtle patterns of what makes an antibody succeed in the lab.
And the third piece sounds like the final quality control check, the system hyperparameters.
Right. This is where all the hard-won experimental knowledge gets encoded into the system. It's a set of rules and thresholds that govern the whole process. How should the generative model search for solutions? How much weight do we give the scoring model? And most importantly, there are hard score thresholds.
Meaning, if a design doesn't meet a certain quality score, it's just thrown out immediately.
Instantly. It doesn't even make it out of the computer. And this is a really crucial point from the sources. This system wasn't just built on public data. The final models, the weights, those thresholds, they were all tuned and selected over more than 50 full experimental design build test cycles.
50 cycles. So they would design something, make it in the lab, test it, see what worked, and feed that knowledge back into refining the AI.
That's the secret sauce. It's this incredibly tight feedback loop. The AI is powerful, but its ability to make things that actually work in the real world comes from being constantly corrected and guided by real experimental data.
So the system spits out, say, a few dozen promising antibody sequences. How do you actually turn that digital file into a potential medicine? Let's walk through that whole de novo pipeline.
The validation is just as important as the design. It's where the rubber meets the road.
It starts with step 1, design specification. The human is still in the loop here. You give the AI the target, and crucially, you have to specify the epitope.
You have to tell it exactly where to bind, a list of specific amino acid residues on the target surface. You're saying, don't just bind anywhere, bind right here.
That's the atomic precision goal. Okay, so the designs are generated. Now they enter the first experimental phase, the binder identification pipeline.
The first question is simple. Does it actually stick?
So you start with library construction, and because JAM is so efficient, you're not making billions of things. It's a small library, right? Maybe a few thousand to a million designs.
A tiny library, by traditional standards. You synthesize the DNA for those designs, you optimize it for yeast, and you clone it into something called a yeast surface display vector.
Which means the yeast cells will now have these computationally designed antibodies sticking out from their surface.
Exactly. And that leads to the sorting and enrichment loop. This is how you find the winners. You take your pool of yeast cells and you mix them with your target antigen. The first pass is usually with something called MACS.
Magnetic activated cell sorting. How does that work?
Think of it like a magnetic fishing net. Your target protein, the antigen, is tagged with a tiny magnetic bead. You mix it in with all the yeast. Any yeast cell that has an antibody that binds to the target now becomes magnetic.
And then you just run the whole soup past a big magnet.
You got it. The magnetic cells, the binders, get pulled out of the solution. It's a way to quickly enrich your population, to go from maybe 1 binder in 1,000,000 to 1 in 100. It's the first big filter.
Okay, so MACS is for bulk enrichment. But then you need more precision, right, to find the very best binders.
Right, and that's where you switch to FACS, or fluorescence-activated cell sorting. This is much more precise. Here, the cells are labeled with fluorescent tags.
So you can measure 2 things at once.
Yes. One tag can tell you how much antibody is on the yeast cell's surface, and another, different colored tag tells you how strongly the antigen is binding to it. A machine then sorts the cells one by one based on their fluorescence. You can tell it, I only want the cells that are glowing brightest green and brightest red, meaning the ones that display a lot of antibody and bind really, really tightly.
And you can do this over and over, each time with less antigen, to select for only the highest affinity binders.
That's the process. And after you've sorted and found your population of winners, you use next-generation sequencing, NGS, to read the DNA from those cells. That tells you precisely which of your original computational designs were the successful ones. The key metric here is the bind rate.
The number of successful binders you found divided by the total number you designed.
Correct. And if that bind rate is high, you're in business. You move to the final stage of characterization.
Step 2, binder characterization. This is where we go from it sticks to it could be a drug.
Right. Now, you produce the best candidates, not in yeast, but in therapeutically relevant mammalian cells, like X by O cells, and you produce them in their final drug formats.
And they tested 2 formats, which I think is important, VHHs and full MABs.
Very important. So VHH-Fc fusions are these smaller single-domain antibodies. They can be great for penetrating tissues or hitting tricky epitopes. Then you have the full monoclonal antibodies, or MABs. That's the classic large Y-shaped format for most antibody drugs today.
So by testing both, they're showing the system isn't a one-trick pony. It can design different types of molecules for different therapeutic needs.
That's the goal. And once you have the purified protein, you do the final analysis. You measure affinity with high precision using something like biolayer interferometry, or BLI. You test its function. Can it neutralize a virus? Can it signal on a cell? And you run a whole panel of early-stage developability tests.
Does it express well? Is it stable? Does it tick to things it shouldn't?
All of that. This whole pipeline is designed to prove that the computational output isn't just an interesting idea. It's a validated, high-quality starting point for a therapeutic program.
Okay, before we jump into the huge performance leap that JAM-2 represents, we need to cover a couple of key technical breakthroughs from the JAM-1 era. One is about compute, and the other is about biology.
Absolutely. These are foundational insights that made JAM-2 possible.
Let's start with the one about the inference process. You made a great analogy to large language models.
It's a very powerful parallel. With LLMs, we've seen that if you give the model more time to think, more computational steps at inference time, it gets much better at complex reasoning. JAM-1 applied that same idea to protein design with a technique they call iterative introspection.
Introspection. So the model is looking inward, so to speak. How does that work for designing an antibody?
Well, in a normal single-shot design, the model gets one chance. It takes the target info and generates the best antibody it can. With introspection, it becomes a multi-round process. The model takes its own best outputs from round 1, the highest scoring sequences and structures, and uses them as a starting point or a condition for the next round of generation.
So it's basically asking itself, okay, this was a good idea. Can I make it even better? Or, based on this good idea, are there other similar ideas I should explore?
That's a perfect way to describe it. It's refining its own hypotheses. And the results they saw when they tested this on the SARS-CoV-2 receptor binding domain, the RBD, were not subtle. They were dramatic.
Just by running 3 rounds instead of 1.
Yes. 3 rounds of introspection with the JAM-1 model led to an 8-fold increase in the number of binders they found at a 100 nanomolar concentration.
An 8x increase just by letting the computer think a bit longer.
But it gets better. Here's the really crucial part. The designs from the single-shot round 1 process, they failed to produce any binders at a 1 nanomolar concentration. Only the designs from rounds 2 and 3 were able to hit that high affinity threshold.
So introspection isn't just finding more solutions, it's finding qualitatively better solutions, higher affinity solutions.
Exactly. And that leads to a really surprising outcome. Your intuition might say that if you're iterating and refining, your solutions should become more similar. You'd expect them to converge on one optimal design.
Right. You'd find one good way to bind and just drill down on it.
That's what they thought too. But the data showed the complete opposite. Iterative introspection actually increased the structural diversity of the solutions.
I have to measure that.
They clustered the designs based on their binding geometry. After round 1, they found only 3 distinct structural clusters. After round 3, that number had exploded to 14 distinct clusters.
Wait, that's counterintuitive. Why would more refinement lead to more variety?
The thinking is that the successful hits from round 1 acted as a kind of a proof of concept for the model. It confirmed that certain binding principles were valid. And with that newfound confidence, the model was then able to explore more unusual peripheral regions of the design space. It discovered entirely new ways of binding that were too complex or non-obvious to find in a single shot.
So introspection isn't just about refining one idea, it's a launch pad for discovering a whole set of new, viable ideas.
It's a catalyst for discovering novelty, and that's a huge insight.
Okay, so that's the compute breakthrough. Let's talk about the biologic one.
Yeah.
JAM-1 also went after a class of targets that are notoriously difficult. multi-pass membrane proteins.
The really tough ones, we're talking about GPCRs like CXCR7 or a protein called Claudin-4. These are incredibly important for diseases like cancer, but they are a nightmare for drug discovery.
Why are they so hard to work with?
Because their natural home is embedded in a cell membrane, they're oily and unstable. If you try to pull them out and purify them for a traditional screening campaign, they often misfold or collapse. You lose the native structure that a drug would need to recognize.
So how did JAM-1 solve this problem before it had the power to go after the native receptor directly?
It used a really clever dual design strategy. It didn't just design the antibody. At the same time, it designed a soluble proxy of the target, a Sol-MPMP.
Soluble proxy, so an artificial, easier-to-work-with version of the target.
Exactly. JAM used its protein design capability to take the native membrane protein and replace the unstable hydrophobic bits that sit in the membrane with a stable, well-behaved, soluble protein scaffold.
But the important part, the part the antibody needs to see, has to be preserved.
That's the genius of it. It preserved the extracellular loops, the bits that stick out of the cell in their native conformation. So you end up with a screening tool that is stable in a test tube but still presents the correct binding sites to the antibody.
And they were able to validate that this proxy was actually a good mimic of the real thing.
They did. For example, their designed Sol-CXCR7 could still bind its natural ligand, SGF1 alpha. And that's a ligand that is known to interact with the entire structure, proving that the AI design core was presenting the binding surface correctly.
And the results against these intractable targets.
It worked. JAM-1 produced the first ever fully computationally designed antibodies against Claudin-4 and CXCR7. They were getting double-digit nanomolar potencies on actual cancer cells.
And what about specificity? That's always a concern with these protein families.
The specificity was outstanding. For the Claudin-4 antibody, they saw more than 100-fold selectivity over other very closely related Claudin proteins, even though the binding regions were 85% identical. So even back in its first generation, JAM was achieving that atomic-level precision you need for a safe and effective therapeutic.
Okay, so JAM-1 was revolutionary. It proved the concept on the hardest targets. But JAM-2 is where this moves from a proof of principle to really an industrial-grade engine.
That's the leap. It's all about reliability, consistency, and just unprecedented affinity right out of the box.
So to test this, they didn't use targets the model was familiar with. They picked a set of 16 completely unseen targets.
Right, a diverse set called Targets at One, a real stress test. And the first headline result is just the raw coverage.
They hit everything.
100% target coverage. They got binders for every single one of the 16 targets. And the success rates are what really stand out, especially when you remember how few designs they're testing.
Give me the number.
The average success rate was 39% for the VHHV format and 18% for the full maps.
Let's just pause on that 39% number. So you're saying if they design, say, 10 VHHs, on average, 4 of them will actually work from a library of just 45 designs per format.
That's what the data shows. In a traditional screening campaign, you'd be thrilled with a 1 in 100,000 hit rate at this stage. This is a completely different world. It means you can basically skip the entire discovery and screening phase and go straight to characterizing your leads. The efficiency gain is just massive.
But it's not just about finding something that sticks. It's about finding something that sticks really well. What of the affinity?
This is the other major milestone. On half of those 16 targets, JAM-2 delivered binders with picomolar or single-digit nanomolar affinity.
Straight out of the computation.
Zero shot. That's the kind of affinity that people spend months or years optimizing for in the lab using traditional methods. Here, it's the starting point.
We have to put this into the context of the broader field, though. There are other AI antibody design platforms out there. How does JAM-2 compare to systems like RFantibody, MBER, or Chi-2?
It really distinguishes itself on, I'd say, 3 main axes: efficiency, the breadth of reliability, and the overall quality of the leads.
Okay, let's take efficiency first. How does it compare to something like RFantibody, which is based on the RF diffusion structure model?
RFantibody is a great system, but it's still playing a numbers game, relatively speaking. You typically need to design and test thousands, sometimes tens of thousands of candidates to get a good binder.
Whereas JAM-2 is working with libraries of dozens.
Exactly. We're talking 45 designs per format. It's a orders of magnitude difference in the experimental effort required. That translates directly to time and money.
What about against the sequence-based models, like MBR or Chi-2? They've also reported some impressive hit rates.
They have, but the key difference seems to be the reliability across a very broad and challenging set of targets. JAM-2 appears to be more of a generalist. The sources point to a direct comparison where they tested against targets that Chai-2 had also reported on.
And what did that comparison show?
It showed that JAM-2 had complementary and broader coverage. It found binders for all 16 of the targets they tested across the two formats. In several cases, JAM-2 succeeded where Chai-2 had reported no binders for a specific target. It suggests that the JAM-2 system is just more robust and less dependent on the specific characteristics of any one target. It generalizes better.
So beyond just finding a binder, the next huge leap for JAM-2 was demonstrating that it can control where it binds. This idea of systematic epitope targeting.
This is moving from discovery to true engineering. This is the functional control piece. For this, they used a second set of targets, target set 2. They took 10 proteins, but for each one, they picked 20 different epitopes.
20 different binding sites on each protein.
And these weren't all easy ones. They were biophysically diverse. Some were charged, some were greasy and hydrophobic, some were in grooves, some were on flat surfaces. They were really stress testing the model's precision.
And the result, could it actually hit these specific user-defined spots?
The result was a massive validation of its precision. For half of the targets in that set, JAM-2 produced binders to between 30 and 70% of the specified epitopes.
That is an incredible level of control. Traditional methods would almost never give you that kind of breadth. They tend to find one or two easy spots on a protein and hit them over and over again.
Exactly. They get stuck on what are called immunodominant epitopes. JAM-2 can systematically survey the entire surface of a protein in a way that was basically impossible before.
Did they learn anything by looking at which epitopes worked and which didn't? Does the model have, for lack of a better word, a preference?
It does, and this provides a really powerful insight into the physics it has learned. The designs that worked best tended to bind to epitopes that had a bit of hydrophobic character and low polarity.
So it likes to bind to the less charged, more oily patches on a protein surface. Why would that be?
It's likely an optimization for stability and energetic favorability. Hydrophobic interactions, the like dissolves like principle, are a very powerful driving force in protein binding. They're reliable.
As opposed to what?
As opposed to complex networks of hydrogen bonds or salt bridges in highly polar charged regions. Those interactions are very sensitive to tiny changes in geometry and are notoriously difficult to model accurately, especially because you have to account for the role of water molecules, which is a huge computational headache. The model seems to have learned that the most reliable path to high affinity and good developability is to maximize these stable, non-polar interactions.
So it's not a limitation necessarily, but an emergent strategy for success that the AI discovered on its own.
I think that's the right way to see it. It's a learned design principle.
This brings us to what might be the biggest biological breakthrough of all: targeting native GPCRs directly on the cell. This just feels like a different level of difficulty.
It's a fundamentally different challenge, and solving it changes the game for a huge class of drug targets.
We talked about how JAM-1 cleverly used a soluble proxy to get around this problem. What was the innovation in JAM-2 that let it just skip that step entirely?
It's really a testament to the increased accuracy of the generative and scoring models. The designs were just good enough right out of the computer to recognize the GPCR and its messy native environment embedded in the cell membrane. So they could target CXCR4 and CXCR7 directly on cells that were overexpressing the full-length native receptor.
And the importance of that is that you're now able to access parts of the protein that are basically invisible in any other context.
You are. You can now access the orthosteric pockets. These are the functionally critical sites. It's where the body's natural molecules, like hormones, bind to turn the receptor on or off. These sites are often cryptic or hidden, and you can't get at them with traditional methods that rely on taking the protein out of the membrane.
So what were the numbers? How well did it actually work against these incredibly difficult native targets?
The on-cell bind rates were, again, remarkably high for such a small number of designs. They saw 11.7% for CXCR4 and 3.8% for CXCR7.
And the affinities were drug-like.
Absolutely. The top designs hit 1.4 nanomolar for CXCR4 and 23 nanomolar for CXCR7, and that's affinity measured directly on the cell surface, which is the most relevant measurement. These are numbers that are as good as or better than what you'd see from a years-long industry campaign.
And of course, the crucial question of specificity. Could they tell the difference between CXCR4 and its close cousin CXCR7?
They could. The antibodies were highly specific. They bound to their intended target and ignored the other, which is exactly what you need to avoid side effects. It's another confirmation of that atomic-level precision.
Okay, let's shift from the biological success to the mathematical proof. It's one thing to say a design works, it's another to prove that it's genuinely new. How did they go about quantifying the novelty of the JAM-2 designs?
They approached it from two angles: sequence novelty and structural novelty.
Sequence novelty seems like the more straightforward one.
It is. You basically take your designed antibody sequence and you run it against a massive database of all known sequences. We're talking over 3 billion sequences from nature, from clinical trials, everything. And you see how different your design is from its closest known relative.
But were they different?
Very. The vast majority of the designs were more than 10% different from anything in that huge database. Many of the VHHs were over 20% different. So the AI is definitely not just repackaging or slightly tweaking things that already exist. It's creating genuinely new protein sequences.
Okay, now for the much harder one. Structural novelty. This requires some pretty intense computational geometry, right? How do you prove that an antibody is binding in a way no one has ever seen before?
It's a really rigorous multi-step process. The goal is to compare the 3D pose, the binding geometry of your designed antibody against all known structures.
So, step one, you need to find all the known antibodies that bind to a similar target.
Right. You take your generated complex and you use the target part of it as a search query against the SABDAP, the Structural Antibody Database. This gives you a list of all existing crystal structures of antibodies bound to targets that are structurally similar to yours.
But similar isn't identical. How do you make sure you're making a fair comparison?
This is the critical filtering step. They use an algorithm called C-align to superimpose the target from your generated complex onto the target from the known structure. And they calculate the root mean squared deviation, the RMSD, between them. If that RMSD is more than 5 angstroms, they throw it out.
Why that filter?
Because if the targets themselves are in very different shapes, then comparing how the antibodies bind to them is meaningless. It's an apples to oranges comparison. This step ensures you're only comparing antibodies that are binding to the exact same conformation of the target.
Okay, so now you have a set of fair comparators. How do you measure the novelty of your antibody's pose?
For the structures that pass that filter, they then calculate the alpha carbon RMSD just between your design antibody and the known antibody without doing any more alignment. This final number tells you how different your antibody's position and orientation is relative to all known solutions for binding to that specific target surface.
And what did this reveal? Did JAM-2 find new ways to bind?
It absolutely did, especially for the less studied targets. For a really common target like the SARS-CoV-2 RBD, the novelty was modest. The RMSDs were maybe 2.5 to 5 angstroms. The model found better versions of known solutions.
But for the hard targets?
For the hard targets with very little existing structural data, the novelty was off the charts. For one target, they were seeing RMSDs between 15 and 25 angstroms. For Claudin-4, it was around 27 angstroms.
A 27 angstrom RMSD is a completely different universe in structural biology. That's not a refinement. That's a fundamentally new binding mode.
It is. It proves that JAM isn't just a fancy search algorithm. It's a true generative system that can invent novel geometric solutions to complex binding problems, especially in areas where human scientists have had little success.
So we have reliability, we have high affinity, we have novelty. But none of that matters if you can't actually manufacture the antibody as a drug. This brings us to the final, critical filter, developability.
This is where so many promising research projects die. A great binder is useless if it clumps together in a vial or costs $1 million a gram to produce.
And to address this, JAM-2 is used to generate what the sources call the largest developability dataset ever for computationally designed antibodies.
They profiled hundreds of their designs against very strict industry standard benchmarks.
Let's walk through the 4 main criteria they used. What's the first one?
The first is expression titer. Basically, how much protein can you make? The bar was set high. A design had to produce at least 50% of the yield of trastuzumab, which is a well-known, highly manufacturable approved drug.
Criterion number 2, monomericity. This is about stability, right?
Yeah.
You don't want your antibodies clumping together.
Exactly. Aggregation is a killer. It can make a drug ineffective or, worse, cause a dangerous immune reaction. So the requirement was that the antibody had to be at least 90% monomeric, meaning single, non-clumped molecules after just one single purification step.
Which means they're inherently stable and don't need a lot of downstream processing.
Right. Third criterion, Polyspecificity. This is the stickiness problem. Does your antibody bind to other random things in the body that it shouldn't?
A major safety concern.
A huge one. They used a few assays for this, and the benchmark was essentially that the designs had to be less sticky than a clinical antibody called bocotuzumab, which is known to be on the high end of what's acceptable.
And the fourth and final criterion.
Hydrophobicity. This is related to the other two. If a molecule has too many greasy hydrophobic patches on its surface, it's more likely to aggregate and be polyspecific. They measured this and compared it against benchmarks like trastuzumab.
Okay, so you have these 4 really tough, non-negotiable hurdles for any would-be drug. What percentage of the computationally designed antibodies passed all 4 of them at the same time?
This is maybe the most stunning number in the whole paper. 57% of all the design antibodies passed all 4 core criteria simultaneously.
57%. That's not a handful of cherry-picked winners. That's more than half of the entire pool.
It's transformative. It means that many of those picomolar binders we talked about already have a lead quality profile. They might not need any further optimization at all. You could, in theory, take them directly into later stage development.
You're talking about chopping years off the typical drug development timeline.
That's the implication. They also confirmed that the designs were very human-like in their sequence, which is important for avoiding an immune response in patients. So the designs are potent, novel, manufacturable, and safe.
So what does all this mean for the big picture? For the drug discovery workflow and for healthcare in general?
The most immediate impact is just a radical acceleration. Think about this. Their workflow for 16 different targets in 2 different formats went from the initial design on the computer to having purified protein with affinity measurements in less than 4 weeks.
With a small team.
With a small team. A traditional approach to tackle that many projects would take a large organization many months, if not years.
And it also seems to address one of the biggest reasons that drugs fail in clinical trials.
It does. A lot of drugs fail because they were over-optimized against an artificial lab target that doesn't really represent the true biology. Because JAM-2 can design directly against the native target on a human cell, you're de-risking the project from day one. You're building for the real biological context, which should hopefully reduce that huge clinical attrition rate.
And looking even further into the future, this level of precision opens the door to genuine functional control.
That's the ultimate goal, not just to design something that binds, but to design something that has a very specific biological effect. You could design an antibody to block a receptor, an antagonist, or one that activates it, an agonist, or even one that pushes it down one signaling pathway but not another. By controlling the exact geometry of the interaction, you can start to engineer function directly. That moves drug discovery from a process of trial and error to a true engineering discipline.
So to wrap up our deep dive today, what we've really seen is this incredible shift from the old way of doing things, this stochastic high-volume screening, to a new paradigm of precise zero-shot computational design.
Exactly. JAM-2 really seems to have proven that generative AI can reliably, and I think that's the key word, reliably produce antibodies that have all the hallmarks of a clinical candidate. Potent affinity, high specificity, genuine novelty, and great developability.
And it's doing this even against the targets that have historically been considered almost impossible, like native GPCRs.
Its ability to use that iterative introspection to find new solutions, its success in systematically hitting specific epitopes, and that incredible 57% pass rate on developability, it all adds up to a system that makes de novo design a practical frontline tool for drug discovery today.
But as you pointed out at the beginning, the success of this whole system was built on a foundation of intense real-world experimental data. Those 50 design-build test cycles that Nabla Bio ran were critical for tuning the models.
They absolutely were. And that brings us to our final provocative thought for you, the listener, to consider. If computational models like JAM-2 keep getting better, if they continue to converge in performance and deliver these high-quality leads so efficiently, how much longer will the bottleneck in this field be the AI? Or will the bottleneck shift? How much longer will deep, consistent, and incredibly expensive experimental data generation, the kind of data that's needed to train and validate these models, remain the ultimate limiting factor for progress in AI-driven therapeutic discovery? At what point does the AI become good enough to learn from its own generated data, and what does that future look like?